All right, so let's further continue our discussion on the VHH and antibody fragments. So for this talk, our focus would be the CMC solution for this different type of antibody fragments. Here is the contents to be covered in this talk. We'll start with some background and application of antibody fragments, followed by the CMC strategy for different type of antibody fragments, including single-chain FV, Fab fragment, as well as the single domain antibody, along with relevant case studies from our platform. So here is a general overview of different applications and advantage of antibody fragments. So as Derek mentioned in his talk, antibody fragments, different from the full-size IgG molecule, their small molecular weight which allows for stronger tissue penetration and enables improved permeability through solid tumors and tissue barriers. And also their simple structure allows for flexible design, which is ideal for engineering for different type of bispecifics, multivalent, or other advanced formats. Last but not least, the small size of antibody fragments are well suited for conjugation applications. In particular, over the last year, we have seen a booming type of applications utilizing different antibody fragments for conjugate applications. In particular, we have seen an increase in use of antibody fragments in in vivo CAR T cell therapies. Basically, the ideal of in vivo CAR T is to deliver the CAR gene utilizing the mRNA LNP structure with the targeted purpose for the antibody and to be delivered in vivo in order to avoid the complex ex vivo treatment and engineering of the CAR cells. So to synthesize the antibody-targeted lipid nanoparticles, there are different general approaches. We could either utilize a one-part type of synthesis, basically putting all the components, including the antibody, mRNA, as well as the lipids all together and through a one-stop synthesis. Or we can do form the LNP, mRNA first, and do the surface conjugation utilizing the antibody. And the last approach that's commonly seen is the post-insertion approach. So we can conjugate the antibody fragment to the lipid and then form the LNP particles later. So in general, this platform has been established by our mRNA CMC team along with the QC and analytical platform. So for this particular talk, we are going to be focusing on the antibody part for the antibody LNP applications, including different type of antibody fragments, such as the single domain antibody VHH, which so far we have seen are the most commonly used type, along with single-chain FV fragment as well as Fab fragment. So first, let's start with the expression system of different antibody fragments. In general, depending on different complexity of these molecules, we can either utilize the CHO cell expression versus the E. coli cell expression. So in general, the E. coli expression allows for a much shorter construction cycle within 2 to 4 weeks, and the expression level could be competitive depending on which type of molecule. For example, if that's a single domain antibody, the titer could be pretty high. And however, there are a number of advantages utilizing CHO cell over the E. coli system. For example, in general, we can secrete the protein utilizing CHO versus a complex lysis process because the protein expressed in the inclusion bodies typically for the E. coli system. And we have to establish a cell bank, including master cell bank and working cell bank, which complies better with regulatory standard requirement compared with E. coli without this establishment of stable cells. And that could also face, when it scale up, face some consistency issue as we scale up the process. And in terms of the product quality, For the CHO-expressed antibody fragments, there's no refolding needed, and it could facilitate certain post-translational modification as well. However, for E. coli, it typically requires the refolding process, and also along with higher level of HCP and endotoxin contamination issues. So in general, the CHO cells enables better GMP compliance, as well as regulatory compliance and good quality consistency across different batches, as well as scale up. Hence, this is a recommended approach for our platform for the production of different antibody fragments. So next, I will go through different strategies for different type of antibody fragments, starting with the Fab fragment. So for the Fab fragment, the typical challenges we have seen during the expression are the sometimes lower expression level compared with IgG, and a lot of times imbalanced chain expression. So we sometimes see overexpression, for example, the light chain, hence the formation of some homodimer species and limited protein stability. Therefore, we optimize our upstream expression platform utilizing a dual vector design and optimization. Keep in mind during the early stage cell cloning, for high assembly of efficiency and optimize upstream feed and temperature control strategy to maximize its expression level. For the downstream purification, because of this molecule's lack of Fc structure, we have to come up with a dedicated affinity platform to capture these antibody fragments. And again, sometimes we see aggregates and homodimer along with fragments impurity present in the cell culture that need to be effectively removed in the downstream process. Additionally, we have some challenges in the analytical development to target specifically for this type of molecules and identify the impurity that are present in the cell culture. So here I'm going to walk you through a case study for a Fab molecule. This molecule we have filed for IND for FDA in the past year, probably 2 years ago. So in general, we haven't seen too much challenge in the upstream. So utilizing our platform process with optimization, the expression titer could reach close to 8 grams per liter. And there are a number of challenges, mostly around downstream and analytical side. So first, as I mentioned, we have to come up with a good capture step without the Fc fragment. And in general, the binding capacity of this molecule on selected resin could be very low. And last but not least, the presence of high level of aggregates and homodimer needs to be removed. So let's start with the selection of the affinity resin. So we systematically screened different type of resins targeting different binding domains. For example, for Fab, we could look for resins that's targeting VL. That's typically protein L, protein L are the most commonly ones that you could think of. Or targeting VH, actually protein A has sometimes good affinity with the VH region. And there are also affinity resins designed to capture the CL and CH1 region respectively. So in this project and overall in this program for the Fab molecules, we actually screen different type of affinity resins, and at the end of the day identified the optimal resin for this project targeting the VL region, and with much higher binding capacity compared with the rest of the resins, therefore successfully reduce the overall production cost for the downstream. And further utilizing this affinity resin, we developed effective method using the elution strategy to remove the majority of aggregates and homodimers, as shown in this slide. So after we pick up the affinity resin, we did a linear gradient elution at different pH and try to see if there's any resolution of our main protein of interest versus the rest of the impurities. As I mentioned, actually, interestingly, for this molecule, the main impurity species are the homodimer. And there are two different types of homodimers, either formed covalently or non-covalently, as we will see from the analytical section later. So during the affinity optimization, we will find different elution pH has a significant impact in separating different impurity with our target antibody. Therefore, we selected the optimal elution pH and successfully separate those impurities utilizing isocratic elution. As you can see that through the AC step, we can achieve close to 100% of the ICC purity with a decent recovery rate over 80%. And we further have two polishing steps to remove the additional impurities such as HCP and HCD. And the overall recovery is at 72%. Two additional points to be highlighted for this molecule. You can see graph on the left-hand side. This molecule is kind of special in the sense that it requires very high SEC purity in order to prevent the toxicity of this molecule. As you can see from this graph, the ELISA binding activity correlates strongly with the high molecular species. As you can see, if we have a high molecular weight species greater at a very low level, even at less than 1%, there's a significant increase in the ELISA binding activity. And this enhanced ELISA binding activity is kind of correlated to the toxic aspect of this molecule. So this brings up very challenging CMC aspects in the sense that we have to tightly control the aggregates level in order to minimize the toxicology of this molecule. So at the end of the day, actually the client asks us to control the ICC level purity to be above 99.5% and apply that in all the GMP production, and we actually did it. So we have actually successfully come up with a robust process that gives the ICC purity above 99.5% and tightly control the aggregates level, and successfully delivered the GMP production for a number of batches. And another interesting aspect about this project is how to quantify the different impurities. So in this case, as I mentioned, the major impurity are the overexpressed lectin homodimers. So actually at the end of the day, we find out that CESDS-NR non-reduced condition is a good way to quantify the different species, because as we isolate the light chain impurities as around the SDS-NR. And as you can see that interestingly, the majority of the species, only a small portion belongs to the LC-LC species, which is formed by the disulfide bond. The majority of the LC-LC impurities are actually being break down at the NR condition because they are non-covalent bonded. So we come to the conclusion and that the majority of the impurity present in this project are the non-covalent bonded light chain homodimer species. Next, let's go through the general strategy for single-chain FV fragment. I think we briefly touched base on the first talk, and I will go into probably give more details on the designing of the single-chain FV in this part. So the single-chain FV molecule fragment, when expressed by itself, whether expressed by itself or combination with IgG to form bispecifics, the number one challenge is always a high level of aggregates that's been present. And also this type of molecule has limited protein stability, meaning that it's prone to form more aggregates at certain pH or ionic conditions. And in some projects, we have seen low expression level for single-chain FV compared with other fragments and antibodies, and sometimes low cell viability during the cell culture stage. So in terms of upstream strategies, similarly, we typically start with trying to optimize the design of the molecule in the beginning, and then choose the optimal clone for high monomer production, and keep the purity in mind in the early stage screening. And by optimizing the feed and cell culture temperature, that could further improve the overall performance of our cell culture. And in terms of the purification, similarly, there's no FC fracture. We have to come up with a good way to capture the single-chain FV fragment, prone to form aggregates, effective solution to remove those aggregates, as well as the poor stability of this type of molecule. So the solutions include screening a suitable capture resin, as well as along with a buffer system and elution condition and additives, utilizing multimodal or mixed mode chromatography to remove aggregates and other aggregate species, and control the protein concentration to avoid the degraded DS quality. So here is one example of a single-chain FV fragment. So as I mentioned, in general, our platform has seen a lower expression level for single-chain FV. For this case, it's 1.6 gram per liter through the cell line process. And during the cell culture, we have identified that the cell viability as well as the productivity of this molecule is heavily impacted by the cell culture temperature. So as you can see, we designed different temperature downshift at 30 versus 33 with 37 degree, and you can see lower temperature at 30 degree significantly help to extend the cell culture duration and save the viability drop. As a result, the titer as well as the QP of the molecule of the cell all improved by 6 times by modulating the different cell culture temperature. And we also considered extend further culture duration from 14 days to 17 days to further improve the expression level. And moving on to the downstream, we designed a protein L capture step to successfully capture this single-chain FV fragment and choose the optimal mixed mode chromatography condition to effectively remove the aggregates. And in this project particularly, it actually involved multiple conjugation steps afterwards, utilizing a C-terminal free cysteine. And we systematically also optimized the conjugation conditions, including the feed ratio, the antibody concentration, reaction pH, etc. And at the end of the day, remove the aggregates by optimizing the AEX and mixed mode chromatography steps. At the end of the day, we achieve a high good product purity over 98% for both SCC and CE, and with overall recovery rate at 65%. So here is another example of the importance of design of single-chain FV component that will impact the stability and developability of a drug. So this case is also a 3-chain asymmetric bispecific antibody format. So initially, the client used certain design of a single-chain FV, which yields in a relatively low titer, as well as a high level of high molecular weights. So to deal with this challenge, I think our antibody engineering team also worked closely with the client to modify the design of the single-chain FV. Basically, by switching the VH and VL domain, we can see the titer increased significantly by 100% from 2 gram per liter to 5 gram per liter, and with a much better improved overall purity of greater than 90%. And this example we have discussed in the previous talk, the introduction of interchain disulfide bond could also have some impact on the stability of single-chain FV fragment. So next, and going to be the focus of this talk, is on the VHH fragment. So here is a comparison of the VHH from camlid versus the human VH structure. So they are similar, but in terms of consisting of 4 frame regions as well as 3 CDR regions. So the main difference between the two different VH versus the VHH molecule lies in first that the VH has 4 highly conserved hydrophobic amino acids in their frame 2 region. And in the VHH, these 4 amino acids are being replaced by hydrophilic amino acids, therefore improve the solubility and stability of the VHH molecule. And in addition, the CDR1 and CDR3, there was introduction of interdomain disulfide bond, which further enhanced the structural stability of VHH compared with human VH. That's why we normally see quite good drug developability utilizing the VHH fragment. So in terms of its CMC challenge, for VHH molecules, sometimes we see lower expression titer, and for some molecules, for a number of VHH tandem format, we have seen lower cell viability. And sometimes the stability, even though VHH has good stability, but VHH combined with other components could yield in a low stability type of molecule format. Therefore, the upstream strategies are kind of comparable to the other counterparts. And for the purification, I'd like to highlight that in addition to, again, no FC structure, we come up with actually a number of different strategies designed for different type of VHH molecules. So we work with a number of different VHH molecule designs. So first one is kind of native VHH. Sometimes it belongs to the VH3 group. So the VH3 VHH actually has quite good affinity to the protein A molecule, protein A ligand, so that we can utilize protein A for capture. The rest of the VHH groups typically don't have any affinity to protein A, so that we have to come up with alternative solution to capture this group of VH. And for further conjugation applications, we have seen different VHH designs sometimes come with unpaired cysteines in their C terminal, so that allows for further conjugation to the LNP or the lipids or other different molecules. And sometimes alternatively, it could be an oligopeptide that allows for a site-specific conjugation using a certain affinity peptide series on the C terminal. And other than the capture challenge, we've seen sometimes HCP removal as well as the formation of dimer, especially when this VHH is designed with unpaired cysteine on their terminal for conjugation. It's not uncommon to see a presence of high level of dimer present. All right, so next we'll go through a number of case studies to tackle the CMC solution for different type of VHH formats. So first group is the VHH with protein A affinity, which is a VH3 group. So this type of molecule is kind of straightforward. So we can utilize protein A. For example, the Prism A resin is commonly used with good affinity with VHH resin as a capture step. And in our platform, we have done a number of cases with just 2-step chromatography to successfully reduce the residual HCP level down to within 100 ppm and with SEC purity greater than 99%. And just bear in mind, because of the small molecular weight, we have to utilize a small molecular weight cutoff ultrafiltration membrane for the UF-DF buffer exchange process. So the second group of molecule also is VHH with protein A affinity. However, these times they have the unpaired cysteines in their design for the conjugation purpose. So here is another example. For the upstream, we can achieve 6.5 gram per liter in their titer. However, the presence of these unpaired cysteines may lead to the formation of the intermolecular disulfide bonds, hence forming the dimers, as you can see. Also from this gel, there's a significant portion of dimers present. So we work with different clients. There are different strategies to deal with this. First, you can certainly remove these dimers through adding the reductants. Typically, with a low concentration of mild reductants, such as cysteine, could break down this type of dimers. Or alternatively, a lot of clients working on this type of applications, because their final application is to utilize this VHH is to conjugate. And the very beginning step of conjugation is the reduction step, utilizing a strong reductant such as TSAB or DTT. So a lot of times they treat this dimer because we understand the mechanism of formation of aggregates pretty well. This dimer is due to the free cysteine formation. So we can also treat the dimer as our intermediate product. And upon the conjugation steps, by adding the reductants, this dimer could be successfully reduced. As we can see, we can either treat dimer as a final product for this DSI, or we can add a small amount of reductants such as cysteine to completely break down this dimer impurities. And in terms of the purification, since it has protein A affinity, we can potentially utilize protein A such as Prism A resin to capture the both monomer and dimer, but relatively low binding capacity and relatively expensive since affinity resin is involved. Alternatively, our platform has also developed our proprietary platform method, and we have in the process of our IP, is to utilize a mixed mode CEX to capture these VHH molecules, all utilizing CEX. So typically, either using CEX resin or mixed mode CEX, we can have a good resolutions between the monomers and dimers, and offering high capacity, binding capacity as well compared with affinity resin. However, if we utilize the mixed mode CEX or CEX as a capture step, we would typically expect higher HCP levels and needs to be further reduced, typically through 3-step chromatography. And the third case, the single dome antibody VHH without protein A affinity. As I mentioned, for this group of molecules, we have our platform method utilizing different CEX-based resin for capture. So here are a number of different cases for different clients, including just the VHH itself or VHH connected in series. So this is actually a bispecific antibody with different binders designed in this VHH tandem. And as you can see, through this platform approach, we can allow for very high SEC purity greater than 98% or 99%. Through the first capture step, and the problem is going to be HCP removal. So we designed a platform 3-step chromatography to deal with the HCP removal problem. And I think this is the last example. This is a VHH in series. So the titer is not bad at 5 grams per liter, but it took us quite a lot of efforts to get here, because initially when we do the cell pool screening, we saw this molecule, probably they are kind of difficult to be folded or expressed, and we've seen a very significant drop of cell viability at the medium screening stage. So we did a careful selection of the combination of different medium with respect to the different single clone, because we see clone and media compatibility could also impact the final condition selected and final viability of the process. And also, similar to the other projects, the cell culture temperature shift plays a significant role in the viability maintenance. As we can see, if we even different temperature shift time This one we first determine the optimal temperature, and shifting the temperature at different days also have some impact on the viability. We cannot do it too early, but if we do it too late, it may impact our titer of impression. So this is basically a balance between the cell growth and the protein production. At the end of the day, we select the optimal temperature shift day at day 7 with the optimal downshift temperature. So moving on to the downstream, we're seeing this molecule is not quite stable, so we see a precipitation occurs during conductivity adjustment when we utilize the mixed mode CEX as capture step. And additionally, HCP removal has some certain challenge. So we did a number of steps, including adding intermediate depth filtration to remove the precipitated HCP through the conductivity adjustment, and add wash 2 in the polishing step to remove additional HCP species. And again, the molecule cannot bear with the low pH condition, so we designed solvent detergent treatment for this molecule, and we've seen low nanofiltration throughput during the nanofiltration stage, and we successfully identified certain metal ion that could, during the formulation development, could help to stabilize this molecule, and it's been added to improve the nanofiltration throughput. As you can see, before adding this additive, the nanofiltration throughput dropped very fast through the experiment, and after the adding metal ion, we successfully maintained much higher throughput for the nanofiltration. At the end of the day, this project delivered over 99% of SEC purity along with the CE purity, with a controllable level of 20 ppm HCP, and with overall recovery of 70%. I would say this is overall challenging project, but we have successfully conducted the talks as well as GMP production, and in the process of, we have submitted IND for this particular case and waiting for the results. So in summary, here through this talk, I'd like to highlight that our platform has quite good experience working with different antibody fragments as well as the antibody AXC. It's a shame that we don't work with ADC, but we work with a number of emerging AXC type of applications. So particularly, we work with a number of pretty well-known companies on the RDC, which is a radionuclide and antibody conjugates, which allows for a targeted radio labeling type of treatment. As well, we are establishing our AOC platform, which stands for antibody oligonucleotide conjugation. This is also an emerging field for targeted oligonucleotide treatment. And along with the previous example I mentioned, the antibody LNP for in vivo CAR-T type of therapy. So our platform covers end-to-end from the early phase discovery to developability study, to naked antibody process development and conjugation process development, formulation development, targeting for this different type of emergent AXC, so antibody plus non-toxic type of conjugates, along with analytical method development and GMP manufacturing for both naked antibody reagent conjugation manufacturing, as well as the fill and finish capability. So the last slide also shows our API service, because for a lot of these antibody fragments, they are not the final DP. So it's a kind of intermediate drug substance, which is going to be utilized for further conjugation to form the final drug modality. So we provide a much faster and more flexible type of service targeting this type of antibody fragments, which also complies with the regular requirement and for different type of applications, whether that's a GMP-like material or it's for the IIT type of application with a much faster turnaround time. So in general, we can deliver the IIT type of material within 3 to 4 months for this different type of antibody and antibody fragments, and 4 to 6 months to get the API materials that meet the IND filing requirements. With that, that brings to the end of my talk, and happy to hear if any questions or feedback. Thank you.
네, 질문 있으신 분은 자리에서 손을 들어주시면 저희가 마이크 전달해 드리도록 하겠습니다. 네, 질문이 따로 없으신 것 같아가지고 네, 여기까지 마치기로 하고요. 혹시라도 따로 질문이 있으시면은 저희 스태프분들께 요청 부탁드리도록 하겠습니다. 네, 감사합니다. 네, 땡큐.